Hello everyone, and welcome to video number 2, in which we're going to be looking at some concepts, and then we're going to be looking at something from phylogenetics called clocks. So let's jump right in and start by looking at some important concepts. Obviously, it's great to define terms, and a sensible one to start off in a series of videos about macroevolution is what the term macroevolution actually means. Well, as general context, we can say that evolution can be studied from two different perspectives. The first is processes that lead to evolutionary change and reproductive isolation within and among populations. Those are the kind of things that we can study when we look out and study what's happening in this particular time slice in the world right now and in experimental studies in labs. But also when we're looking at fossils, say, we can study the long-term fate of species or higher ranked taxa through geological time. The question really that we have to ask ourselves is, are those two just aspects of how we choose to study the process of evolution, or are there meaningful differences between the processes that are occurring on those two timescales? More correctly, perhaps, we could ask, is macroevolution a thing? Well, exact definitions of macroevolution differ, both as conceived today and in the past. And indeed, there's a paper on how best we may decide to define macroevolution, which I've placed on this slide here for you. And this argues that there are three major different definitions. Number one is that macroevolution is the evolution of taxa of the rank above species in the Linnaean taxonomy. Definition number two is that macroevolution is evolution on a grand timescale. Definition number three is that macroevolution is evolution that is guided by the sorting of variation between species, as opposed to the sorting of variation within species, which we would call microevolution. Hauptmann 2020, the paper I've referenced on the slide, makes the case that only definition number three allows for a consistent separation of macroevolution from microevolution. Certainly, definition number one is difficult because we can't really identify meaningful differences between the evolution of species and between the evolution of ranks higher than species in the Linnaean taxonomy, possibly reflecting the fact that the ranks higher are to an extent arbitrary. And the second definition is quite vague. How do you define grand within this framework? That leaves us with definition number three, where essentially in this world, speciation would be separating macroevolution from microevolution, and macroevolution would occur through differences in speciation and extinction rates. Given that we're left with that definition, let's jump in and dig in a little bit deeper to what that definition says. The question we may be asking ourselves here is that a useful distinction to be making? Well, as we have highlighted throughout this course, evolution on a large scale is indisputably the accumulation of smaller changes and changes at smaller scales. But the question really about this definition is whether that is a useful one to make. Is the impact of processes such as fitness, competition, and selection different at the two levels that we're talking about? And if so, then actually making a distinction to allow us to kind of to provide a framework to our study is a useful thing to do. The paper that I've cited makes the case that there is a meaningful difference, and thus macroevolution is a useful concept, which one where a macroevolution reflects the sorting of inter rather than intra-specific variation. To illustrate this, let's look at this graph on the left-hand side here. This shows a situation where we have speed of a prey species on the x-axis and the frequency of that species on the y-axis, i.e. how many of those things are alive. These are prey species A and B. We can then imagine that we have a predator, and let's say, for argument's sake, if we have a predator, number one, that can run as fast as is dictated by this arrow on the graph here, then this will elicit a microevolutionary response in prey species A, because some of these individuals, those to the right-hand side of this speed here, can escape from that predator. And so that predator will ultimately eventually shift the distribution of speeds of prey species A towards the right on this graph. That, however, is not true of predator species number 2 here, where this will be able to catch all of prey species A if it preys upon them. So it won't be able to elicit a microevolutionary response in prey species A. However, predator species number 2 here also won't be able to capture prey species B that's shown on the right-hand side of the graph here, and thus we could expect there is a macroevolutionary difference between prey species A and prey species B in this situation, given the two predators that we have shown in this graph, suggesting that there is this meaningful difference between inter and intra-specific variation. Okay, so in this framework, then we can say that, okay, microevolution and macroevolution both respond to an environment, but within species variation, intraspecific competition will be in the realm of microevolution. If that leads to changes within a population, that will be anagenetic change. That's change whereby the entire population shifts, which we've learned about elsewhere in this course. However, if that leads to a speciation event, that intraspecific competition can lead to a separation of populations, sometimes because of geographic barriers, then we may expect those new species to be then further differentiated by between species variation, and that will lead to the evolution of different clades. Okay, so that I think is possibly a good way to think about this distinction between micro and macroevolution, even though these are the accumulation of small changes. There is on the basis of this argument, a meaningful difference between the two, and so we may want to consider that as a thing. But do be wary of kind of narratives that take this distinction between micro and macroevolution to suggest that we can observe microevolution, but that macroevolution isn't a thing. This is a typical creationist argument, which doesn't actually hold much water. It's not very kind of robust to the evidence, because actually we see evidence of macroevolutionary change throughout the tree of life. So that's macro versus microevolution. And now, when writing this course, I had to pick very carefully what concepts I featured, especially in this lecture, because there's a whole host of possibilities. And I chose to talk a tiny bit more about the Red Queen hypothesis, because I think it's a super duper interesting one. We met this before in the maintenance of sexual reproduction in the milestones in evolution material on this course. So the origin of the Red Queen comes from Alice Through the Looking Glass, when the Red Queen says, you see here, it takes all the running you can do to keep in the same place. If you want to get somewhere else, you must run at least twice as fast as that. The idea of the Red Queen was originally developed in the 1970s in paleontology, but over the years, it has come to encompass numerous evolutionary concepts and theories that champion biotic interactions, that's interactions between living things, as a significant mechanism for evolutionary change. There's quite a lot of baggage about this, lots of definitions of what the Red Queen may or may not be. And the paper I've cited by Strutz et al. From 2018 on this slide unpacks that really nicely. But I think a really good illustration of the difference between the Red Queen and the court jester type thinking is shown in these graphs in the middle from that Strutz paper. Now this shows, these graphs show an important trait that matters to both predator and prey, i.e. speed. In the top graph here, we have a blue species that is going extinct. And if this blue, let's say that's a predator, it may go extinct because its prey, the green species, evolves to be able to avoid predation here. So you can see that looking at the speed of this prey here, this green line, it may evolve to get faster and faster due to a microevolutionary change imposed upon it by predator, the predator that is this blue species here, in one adaptive zone. And we can then imagine that, okay, if this prey evolves faster and faster speeds and this predator is not able to keep up, eventually we may expect the predator species to go extinct as a result of a Red Queen interaction. That may, of course, take our prey into another adaptive zone where actually it's preyed upon by a different predator. But nevertheless, that's a red queen type dynamic that leads to the extinction of a species. In contrast to that, possibly on different time and spatial scales, we could imagine a world where actually everything occurs within a single adaptive zone, but maybe at this scale, abiotic change leads to the extinction of our blue species, but at the same time, that could lead to the evolution of a different species, a different prey species that's shown in orange here. And so the species of this predator is driven by abiotic change, and the species turnover that we see is abiotic in nature, is not caused by competition. And all of this continues through time with these predator and prey species interacting with some species turnover happening there. The graph shown on the right from the same paper tries to unpack this impact of scale a little bit further. So here you can see two interacting co-evolving species, A and B. They may, over time, look like they have a constant relative fitness. There aren't changes in their fitness through time on a big scale. But if we actually zoom in a small bit of this, if we imagine one of these is an antagonist, like a predator, and the other one is prey, they may be evolving out of sync with each other. When you have more predators, then you have less prey. That means they can support fewer predators, and that means that actually you've got this constant co-evolution between those two species through time. Yet, as we look at that on a big scale, we may imagine that that is actually that looks very much like stasis. And even if we dig into one of these species, we may see the same dynamic occurring within populations of those species. And so we've got this situation here where we can actually imagine over a long time we don't see much change, but there is constant change occurring underneath at this finer scale. And of course, this relative fitness could then be impacted by significant abiotic events, altering the balance of the interaction of these two species. And so really what we're looking at is this fine scale red queen type dynamic that's being overprinted on bigger timescales by a chord Chester idea. I think this is a really interesting idea. It's worth noting that an implication of this last example is that the hypotheses have different evolutionary and spatial scales. And that's an idea that has been around for a while. And the paper that I cite here from 2009 provides an overview of that formulation from the world of paleontology. It suggests that competition, predation, and other biotic factors shape ecosystems locally and over short time spans. So when we're looking into the fossil record and we're looking at hundreds of thousands of years to millions of years at individual localities, that's places in the world that aren't that extensive, we may see red queen type dynamics. However, extrinsic factors such as climate and oceanographic and tectonic events such as mountain building and continental collisions may shape larger scale patterns. So that's both regionally and up to a continental scale and globally through thousands and millions of years. So on this small scale, you've got the red queen. On this big scale, you've got the court jester. This also highlights that at really, really big scales, you may actually have abiotic factors that are influencing species that kind of cancel each other out, therefore giving an apparent red queen scale effect. If you want to get some more a clearer idea of how all of this slots together, this is a really, I think, nice paper to read. I also wanted to highlight, though, that the second paper here which is a recent paper that does an analysis of the Phanerozoic, so from the Cambrian through to today, marine animal fossil record. And that paper suggests that the origination and extinction of genera may be controlled by climate on timescales that are less than 40 million years, but biotic effects on longer ones. And so actually, there's still a lot of ongoing debate about the nature of scaling in the red queen, which is going to be an exciting place for future research in the next few years. I wanted to finish by highlighting that many interesting questions in evolution surround the rate at which evolution occurs. And actually, in the last 40 or 50 years, we've developed a suite of tools that help us understand rates of evolution. Now, we've mentioned elsewhere in this course how we can build evolutionary trees, phylogenies in principle, from both DNA and morphological data. And this is just adding a bit more detail to that picture. And I wanted to add the concept of a thing called a clock. We can do this with molecules, DNA, and we can do this with morphology. Either way, the principle behind this is relatively simple. I'm not going to explain in detail how we do it, because that, when I did that, made my video way too long. But in principle, actually, I hope it's relatively understandable, and it's shown by this diagram here. If we imagine we only really have the DNA of living species, but we can, to use DNA as an example, get the DNA of two species here, and we could code that DNA and we could see that, okay, these species differ by 4 base pairs, okay? Then we could also find a fossil that we believe represents the common ancestor to those 2 species. And through that fossil and the geological column and ultimately radiometric dating, we may know that fossil is that common ancestor dates from 50 million years ago. Because we have that fossil and we have its date, we can say that, okay, that means that 4 base pairs is the amount of change we would expect between 2 lineages over 50 million years. We could then extrapolate that and say, for example, that after 25 million years of evolution, since those 2 lineages split, we would expect 1 base pair different. That is a rate of change of the DNA, and that rate of change will allow us to do to date any 2 splits on any evolutionary tree. Even though I've only on this example included 2 living species, we could do that with any number of living species and any number of common ancestors, which we can call calibration points on this tree. And this allows us to recover rates of evolution across huge swathes of time, depending on which organisms we select to be in our tree and which calibration points we select for an analysis. I hope that kind of makes sense. That's the broad picture of how we do a clock-based analysis. There's obviously a lot of technical and methodological detail underlying that. When I recorded the video with details, it was too long, but as I said, to make up for that, if you want to learn a tiny bit more about it, you're always welcome to ask me in person. But also at the bottom of this website is an article I wrote that tries to explain how that works in a tiny bit more detail for you. So with that, that brings me to the end of this video, and I'll see you shortly in video number 3. See you there.